All right, ladies and gentlemen, welcome back. We are going to continue our discussion on Hardy-Weinberg by really starting to get into some examples of the math itself. So when we look at the Hardy-Weinberg principle, it is a mathematical model. So when we look at this, we have these two big equations that exist. The P plus Q equals 1, and then the P squared plus the 2PQ plus the Q squared equals 1. So we need to understand when we're looking at the genetic situation here, what do those variables actually represent? So when we talk about your P, this is your frequency for your dominant allele. So when we look at the way that we would typically designate this, it would usually be some sort of capital letter. So let's just use A's for our purpose here. So the frequency of that dominant allele is your P. When I look at my Q's, then Q's are your recessives, okay, or your lowercase letters. So if you think about it, the frequency of those two has to equal 1 or 100% because I'm taking every dominant allele present in my population, every recessive allele present in my population for that given trait. And if I've counted everybody in the population, then I should have that equal 1 or 100%. When I come over here to the P squared, the 2PQ, and the Q squared, once again, I'm taking frequencies for the collective gene pool for every individual for that trait in that population. So again, it should make sense that that equals 1 or 100%. But now, instead of just looking at the allele frequencies of either dominant versus recessive, now I want to know the genotypic frequencies. So when I look at my P squared, this is for that homozygous dominant. And if we keep using those A's as examples, this would be somebody who would have a genotype with 2 capitals. If I look at somebody who's 2PQ, now I'm talking about somebody who is heterozygous. Okay, so they have 1 of each. And if I come down here to Q squared, these guys are my homozygous recessives. And so they would have 2 lowercase letters. All right. And keep in mind when we look at this, right, if I am homozygous dominant, that means I show the dominant trait. If I'm heterozygous, assuming this is traditional Mendelian genetics, pea plants, for example, right, then I would still show the dominant trait, right? So if I still look at these guys, this is still dominant in terms of its phenotype. Okay. These guys are also dominant phenotype. These guys then would be the recessive phenotype. So let's take a look at what this actually looks like in practice of a real example. So let's talk about sickle cell disease because this is an interesting example of how traits get passed on differently in different populations. So sickle cell disease is due to the homozygous presence of the sickle cell allele. All right, and so we're going to use this symbol HBS, right? Because it is your hemoglobin and it's sickled. It's got a sickled shape to it. Your heterozygote individuals have 1 of each. So they have a sickle cell allele and then they have a normal folding allele as well. So having a sickle cell trait that is identified by a blood type, what we find is about 0.25% of African Americans, so Americans living here in the US that are of African descent, are homozygous and have that sickle cell disease, right? So they're homozygous recessive, okay? In certain parts of Africa, on the other hand, there is about a 4% frequency for the sickle cell disease. The Hardy-Weinberg model enables us to predict when the frequencies in a population will remain constant Because people with sickle cell disease frequently die in childhood, the frequency of the alleles causing this disease should not remain constant. Each death removes a pair of sickle cell causing alleles from the population. The locations in Africa where the frequency of the sickle cell form is highest, so the HBS, there is a fatal form of malaria present as well. Studies reveal that people homozygous for the normal allele often die of malaria. However, people with the heterozygous genotype do not contract the fatal form of malaria. So let's break down what this means. So if I look at my genotypes, right, I have three options. I can either be HBA, HBA, right? So when that comes down to, do I have sickle cell? No. Can I contract malaria? Yes. Okay. This is my homozygous dominant, so my Hardy-Weinberg variable here is the p squared. If I look at another option, I can be heterozygous for this. Okay. Do I have sickle cell? No, although we'll talk when we get to genetics about the fact that this person is a carrier for it. All right, so they can pass it on to their offspring, even though they don't show it themselves. Do they contract the malaria though? No, which is an interesting piece to this. And when we look at your Hardy-Weinberg variables, this is your 2PQ. All right. Third option, I can be homozygous recessive, in which case I've got two copies of that sickle cell, so I definitely show the sickle cell disease, but I don't contract the malaria. All right, having at least one copy of that sickle cell provides some protection against that malaria. I come down to my Hardy-Weinberg variables, and that's my Q squared. So when we look at actually doing the math for this, something to keep in mind is the fact that being homozygous dominant or heterozygous, I can't look directly at you and be able to tell you from your phenotype, your physical expression of your genes, whether or not you are homozygous dominant or heterozygous for it. Do you carry two traits or two alleles for that trait or only one? I can't tell just by looking at you because all I see is your physical manifestation of that trait. So when we look at calculating Hardy-Weinberg, we want to try to start with homozygous recessive because that is the genotype that I can know for sure just by looking at your phenotype or your physical expression. I know that you have to have two recessive copies in your genotype because you're showing the physical recessive trait. Hopefully that makes sense. So when we look at sickle cell, right, let's start off with the African American population. And we were told that it was 0.25%. When we work in Hardy-Weinberg, we always convert those to decimals as opposed to percentages. So that means that I needed to move this decimal place 2 spots, right, to convert that percentage into a decimal. So my Q squared, right, my homozygous dominant, or excuse me, homozygous recessive, these are the people with the sickle cell disease. The frequency of being homozygous recessive for sickle cell is 0.0025. Now I want to know what the frequencies for being homozygous dominant versus heterozygous are in my population. So I'm going to start off with that homozygous recessive, and I need to backtrack this. I need to get Q by itself so that I can use that formula of P plus Q equals 1 to then figure out the P. So my Q, if I take the square root of it, that gets rid of, by doing this, that gets rid of the square, right? So that I can get Q by itself. So if I do that to the number, what do I end up with? So when I do this and I get Q by itself and I go ahead and I take the square root of this number, right, then Q ends up equaling 0.05. Okay, so I now know what Q is. That's going to now allow me to figure out what P is. So now that we know that q equals 0.05, I can now utilize that p plus q equals 1 equation to figure out what p is. So if I have that p plus q equals 1, remember your simple algebra. So I ultimately want to get p by itself. So if I subtract q from both sides, then they cancel out over here, and that leaves me with p equals 1 minus q. So now I can take what I have for q and plug that in. So p equals 1 minus 0.05, and that's going to give me that 0.95. Okay, so now I want to figure out, based on the fact that I know that p equals 0.95 and q equals 0.05, now I can figure out what the frequencies for each of the corresponding genotypes would be. So if I look at my homozygous dominant, remember that's my p squared, right? My heterozygous is my 2pq, and my homozygous recessive is my q squared. So now I'm going to plug in these values to be able to calculate the frequencies. So if I know that my p is 0.95 and I square it, then the frequency of the homozygous dominant is 0.9025, or approximately 90%. My heterozygotes are the 2 times the 0.95 that I have for p times the 0.05 that I have for q. If I calculate that, then I get 0.095, or about 9.5%. Come down to the q, 0.05 squared gets me back to that 0.0025 that I started with. So that's a nice double check just to make sure you've done all your math correctly. And if you add all of these up, they should equal 100%, or at least close to it, depending on if you end up needing to round anything significantly somewhere along the way.